So the next three chapters, 7, 8, 9, will be on the nervous system. And today, right now in chapter 7, we'll just be talking about neurons. And neurons are just the typical nerve cell in the nervous system. So we'll look at general cells of the nervous system, the structure of neurons, and then a little bit how nerve impulses travel throughout these nerve cells. So the overview of the nervous system, its function is to integrate and coordinate the body's activities. We divide it into your central nervous system, which is your brain and spinal cord, and your peripheral nervous system, which is your nervous tissue outside of your brain and spinal cord. So your peripheral nervous system would include all of your spinal nerves and nerves that are leaving your brain and spinal cord. There's two types of specialized cells in the nervous system. We have neurons and neuroglial cells. The neurons are nerve cells. They make up the majority of these specialized cells, and the neuroglial cells just work to support those neurons. So neurons are excitable cells that generate and transmit messages from the brain to the spinal cord to some sort of target cell in your body. And neuroglial cells, also sometimes we just call them glial cells. They actually outnumber neurons 10 to 1, and there's several types, each with a specific function, and they provide support, growth factors, and insulating sheaths around the axons of neurons. There's three categories of neurons. We have sensory or afferent neurons, which bring sensory information to the brain and spinal cord. Motor efferent neurons bring motor transmission or motor directions to target tissues. And interneurons or association neurons are just interneurons are neurons between the two. So sensory neurons and motor neurons, you can see there what they do. Interneurons are found only in the brain and spinal cord. They're between the two sensory motor neurons. They account for about 99% of the body's neurons. So here's a look at an example of a sensory neuron would sense pain or sense some sort of stimulus, whether it's the light that you're seeing right now as you're watching this video. And the sensory receptor would sense that pain and send that impulse or that signal through the axon of a neuron, the cell body, through an interneuron, and the interneuron would connect a sensory neuron to a motor neuron in the brain or spinal cord. And the motor neuron will then send out the response to usually a muscle. So this example is a common pain reflex response where if you touch something that's hot, the motor neuron will tell your muscle or your finger to pull away and back away from it. So sensory neurons carry impulses to the brain and spinal cord based on something that they perceive from the external or internal environment. And motor neuron is the effector neuron. So it'll act on muscles, glands, other body tissues to usually have some sort of effect that was perceived. The shape of a typical neuron is specialized for communicating with other cells. We have dendrites, axon, and a cell body are our three main parts of the neuron. Dendrites receive signals from other cells. They carry the information towards the cell body. The axon carries information away from the cell body to either another neuron or an effector organ or muscle. And the cell body itself will have a nucleus, other organelles, and functions to maintain the neuron. So this is a great picture of a neuron. The dendrites receive information from other neurons or from the environment. Here's your cell body with the nucleus that controls the cell's metabolic activities. And here is the axon, the elongated portion, which conducts the nerve impulse away from the body. The axon will either connect to another neuron or a muscle or a gland. And the axon endings will release chemicals called neurotransmitters that will get passed on to the next neuron to continue passing that signal along. So nerves consist of parallel axons, dendrites, or both from many neurons put together. So we name nerves in your body. They're covered with a tough connective tissue, and all they are a bunch of parallel axons and dendrites put together. They can be classified as a sensory, motor, or mixed nerve, depending on the type of neurons they contain. And many axons will be covered with what we call a myelin sheath. This is found on most axons outside the central nervous system and some within the central nervous system. And again, the central nervous system means your brain and spinal cord. It provides, the myelin sheath provides electrical insulation that increases the rate of conduction of a nerve impulse, and it's composed of the plasma membranes of glial cells. In your peripheral nervous system, Schwann cells form the myelin sheath, and there are little gaps between adjacent Schwann cells called the nodes of Ranvier. And the messages will travel faster as they jump from one node of Ranvier to the next in a type of transmission we call saltatory conduction. So here's a look at the axon being wrapped with this myelin sheath. And again, this is showing the nucleus of the Schwann cell that creates the myelin sheath. And it's just basically wrapping around the axon like electrical tape. Here is showing saltatory conduction, how the nerve impulse will jump between myelin sheaths in these nodes of Ranvier. And the nodes of Ranvier are the spaces between myelin sheaths. Multiple sclerosis is a disease in which the myelin sheaths in the brain and spinal cord are progressively destroyed over time. It results from the destruction of the myelin sheath that surrounds axons in your central nervous system. The resulting scars, scleroses, and can interfere with the transmission of nerve impulses, and it can result in paralysis and loss of sensation in your limbs, including your loss of vision. For some reason, MS most often, not always, affects women in their late 30s, early 40s. It's a progressive disease. It gets worse slowly with time. There's no cure for it. You can just manage it, manage the pain. Here's a great video on the structure of a neuron that I would encourage you to watch. Again, take a look at my PowerPoint file in the modules. So a nerve impulse or an action potential, it's an electrochemical signal involving sodium and potassium ions that cross the cell membrane of the axon through ion channels. Each ion channel is designed to allow only certain ions to pass through it, and these ion channels can be permanently opened or regulated by a gate, and the transport does not require any energy as the ions follow a concentration gradient, which means they're traveling from an area of high concentration to an area of low concentration. These ions are also transported across the membrane by the sodium-potassium pump, and this pump uses cellular energy, or ATP, to eject sodium ions from within the cell and bring potassium ions back into the cell. When a neuron is not conducting any sort of nerve impulse, we say it's resting. And the resting potential is the inner surface of the axon membrane. And we label that in millivolts, so a measure of electrical activity, and that's at about -70 from the outside surface. At resting potential, there are more sodium ions outside the membrane, and there are more potassium ions inside the membrane. And this state of more sodium outside and more potassium inside is maintained by the sodium-potassium pump. An action potential is when a neuron gets stimulated by some sort of stimulus, whether that's pain or light or any sort of stimulus that would, give this neuron a shot of electrical activity. There's a sudden reversal of charge across the membrane because sodium gates open and sodium ions enter the cell. After that, potassium gates will open and potassium ions will rush out of the cell. And this change in reversal of charge across the axon membrane is what passes on a signal down the neuron. We call threshold the minimum charge that causes your sodium gates to open. Depolarization is the reduction of charge difference across the membrane, and repolarization is the restoration of charge back to resting across the membrane. And these changes can occur in a wave along the axon. So here we have an axon showing the sodium in purple gate and potassium gate in blue. The loss of charge difference across the membrane, depolarization occurs as sodium ions enter the axon. So this would be an action potential, the start of depolarization as sodium ions enter the axon, and then return of membrane potential occurs when potassium ions exit the axon. So this is a basic graph of action potentials. You can see resting potential here, always at -70 of an axon. When sodium ions enter the neuron, because they're positive, it brings the inside of the neuron to a more positive number, and we call this the action potential. Potassium ions will leave the neuron, and when positive charges, because potassium is also positive, when potassium ions leave the neuron, the inside of the axon becomes more negative again. Threshold is just the point at what we call the amount of charge difference needed in order to establish an action potential or a passage of this signal. Once started, action potentials do not diminish. They do not vary in intensity with any sort of difference in the strength of the stimulus. So we call it an all or nothing response. And for a brief period following an action potential, the neuron can't be stimulated again. And we call that the refractory period. And that occurs because your sodium channels, which are the first ones to open, take about 1 to 2 milliseconds before they can be reopened again. Here's a great video on action potentials and nerve impulses for you to watch too on your own. Synaptic transmission, this is communication between a neuron and an adjacent cell, and it occurs by what we call neurotransmitters. The synapse is the junction between the neuron and another cell, and the synaptic cleft is just the gap between the two cells. Neurotransmitters will diffuse across this gap to continue the transport of that signal. And the synaptic knob is a swelling at the end of the axon of the presynaptic neuron. So here's a look at the synaptic knob. The synaptic vesicles will contain neurotransmitters that will be released in the cause of an action potential traveling down this axon. The neurotransmitters will be released in the synaptic cleft, which is the space between the two, and they will bind to these receptors, which will cause ion channels to open, which will allow the signal to continue passing. The nerve impulse reaches the synaptic knob. Calcium ions move into the synaptic knob, which releases neurotransmitters into the synaptic cleft, that space. And the neurotransmitter will diffuse across that cleft, the space, and bind with receptors on the membrane of the postsynaptic neuron, causing an ion channel to open. You can see that is depicted here. At an excitatory synapse, binding of the neurotransmitter to the receptor causes sodium channels to open, increasing the likelihood that another action potential will occur in the postsynaptic neuron and continue the signal passage. At an inhibitory synapse, binding of the neurotransmitter to the receptor opens different ion channels, and the interior becomes more negatively charged than usual, trying to reduce the likelihood of reaching an action potential. A neuron may have as many as 10,000 synapses with other neurons at the same time. Some synapses can be excitatory or inhibitory, and summation is the combined effects of the excitatory and inhibitory effects at any given moment. It can determine whether an action potential is generated, and this level of integration provides very fine control over all of your neuronal responses. So here's a look at a cell body receiving signal from many different axons at the same time. Some are excitatory and some are inhibitory. Temporary effects of synaptic transmission. Once released into a synapse, the neurotransmitters are quickly removed. Some are deactivated by enzymes and others are pumped back into the synaptic knob of your presynaptic axon. And that's to control the amount of neurotransmitters that are moving on. Here's a great video about synaptic transmission. There are dozens of neurotransmitters that get released from the presynaptic knob. Some neurotransmitters produce different effects on different types of cells. Acetylcholine acts in both your peripheral and central nervous system, and it's released at every neuromuscular junction, which is the junction between a neuron and a muscle cell. Myasthenia gravis is an autoimmune disease. Remember, whenever you see the word autoimmune, that means the body attacks itself. And this disease is where it attacks acetylcholine receptors at the neuromuscular junction, resulting in little muscle strength. So this disease would attack receptors in your muscle cells, so they wouldn't be able to receive the impulse from the neurons that are trying to get them to move. In your central nervous system, different neurotransmitters are associated with different behavioral systems. Norepinephrine regulates these things. Serotonin promotes a feeling of well-being, and dopamine regulates emotions and complex movements. Changes in the levels of neurotransmitters cause disorders. Alzheimer's disease is associated with decreased levels of acetylcholine. Clinical depression can be with decreased levels of serotonin, dopamine, and norepinephrine. And Parkinson's disease can be associated with decreased levels of dopamine. So after this chapter, you should be able to know these. So just, again, use this slide at the end of the chapter as a checklist to make sure you're getting all the required information out of the slide. Thanks for listening.